Hello, everyone. My name is Yimin, and the title of my PhD project is Structural Basis of Human Melanogenic Enzymes. And so for my project, I have my supervisors here at the ESRF, and my industrial partner is based at the InnovaXN. And also I'm registered at the UGA as their PhD student. So to start off, I would like to give you some really basic information of background of my project. So melanogenesis can also be translated into biochemical process of melanin production and distribution in human. And here we are particularly interested in melanogenesis in human. So in human, we have two types of melanin, eumelanin and phenylalanine. And these are natural pigments that we can find in human skin, hair, and eyes, and it makes us different. So for this process, it is tightly regulated by three main enzymes, which are tyrosinase, TYR, tyrosinase-related tyrosinase-related protein 1, TYP1, tyrosinase-related protein 2, TYP2. And I have put 1, 2, and 3 here so that you can refer to them later on a later slide. So the human melanogenesis involves tight communication between the keratinocyte and melanocyte in our skin. And in very general word, when we have, let's say, exposure to UV because of the sun, the keratinocyte will produce a chemical that will be sent to melanocyte. And this will trigger melanogenesis to occur, and which will then lead to the production of this melanin, and this will be sent back to the keratinocyte so that it can give us protection. So if we take a deeper look into this process of melanogenesis, so it involves two pathways which both start with tyrosine, and then we will end up either with melanin or phenylalanine. But for my project, I am more interested in melanogenesis because this is where our enzymes are involved in. And as you can see, so number one, the tyrosine, they are needed to kickstart the melanogenesis pathway. And as for the second protein, it is believed that this TYP1 protein might be involved in this particular step, but it is still a kind of a part where we need to investigate because there are mixed information on the literature. As for TYP2, the third protein, it is known to convert this dopachrome and to DCA. So you might be wondering, why are we interested in doing this project? So we are kind of hoping that with this project, we will combine biology, structural biology, and chemistry so that we can study the 3D structure of all these enzymes. And so at the moment, we know the structure of one of these proteins for sure. And it is published already by a previous student in our group. And as for the other two proteins, we still don't really know how they look like. And for tyrosine, the first protein, for example, we do have some structure, but they are not of human origin. So it would be really exciting for us to find out how it looks in human. And as for the second part, through interacting substrate. So if you remember, because we are still unsure whether where these three proteins interact, especially for TYP1, the second protein. So therefore, we are highly interested in carrying out this experiment with our industrial partner at InnovaXN so that we can find out what is the real substrate for the human melanogenesis pathway. And all these, we will hope then we can use the information that we find for drug discovery and development project. And at the moment, we have made great progress with the protein expression and purification. And we also are testing some chemicals that will were given by our, produced and given to us by our partners, collaborators at the UGA. So the outcome is quite interesting, and I'm not going to reveal too much because there might be some confidential reason. So yeah, stay tuned for our publication soon, maybe. And yeah, that's the end of my slide. Thank you.